Stay away from the negative people, as has been said by the Einstein. They have a problem for every situation. So coming to the ninth parameter, the enzymes, many bacteria produce tissue degrading enzymes which are coagulase, forms blood clot produced by the Staph aureus, works with serum factors to coagulate plasma, so it forms fibrin walls around staphylococci lesions to protect the bacteria from destruction. Collagenase, the name is explanatory, breaks down collagen, major protein of fibrous connective tissue, and promotes spread of the infection into the tissues. Hyaluronidases breaks down the hyaluronic acid, a polysaccharide that holds together connective tissue. So connective tissue destruction, and so bacteria just spread through the tissues. Here one can see the epithelial cells and the bacterium through the enzyme hyaluronidase. They are capable of getting entry into this tissue, and also here the collagenase works to destroy the collagen fibers. Here the hyaluronic acid is destroyed, and then the end result is the entry of the bacteria into the deeper tissues, and these are invaded. Here again one can see the outside, and the bacteria using the enzymes hyaluronidase and collagenase getting inside the tissues, and they are capable of causing the deeper infection. And enzymes used for the penetration, again one can see the coagulase that helps to protect the bacteria. So these are staphylococcus bacteria. They are protected from degradation by the complement and the other system. And here again you can see the hyaluronidase and the collagenase, which make the safe way for the bacteria to get enter into the tissue and to the deeper tissue. So these enzymes just destroy the barrier which herald the entry of the bacteria. So the enzymes are used for the penetration. And examples are Clostridium perfringens, some staphylococcus, and anaerobes. Then the enzyme is lecithinase. The other enzyme is streptokinase. Fibrinolysin breaks down the fibrin and dissolves the blood clots formed by the body to isolate infection, so the infection can spread by dissolving the clot produced by the many hemolytic streptococci. It activates the proteolytic enzyme of the plasma. It dissolves coagulated plasma and helps in the spread of the streptococci through the tissues. Used in the treatment of the acute myocardial infarction as SK to dissolve fibrin clots. The next enzyme is cytolysins, hemolysins, and leukocidins. As their names are explanatory, they destroy the leukocyte polymorphs and macrophages, so prevent phagocytosis by these cells. IgA proteases, anti-immunoglobulin protease, it helps survival on the external surfaces being produced by the Haemophilus influenzae, Strep pneumoniae, Neisseria gonorrhoeae, Neisseria meningitidis, and Pseudomonas. The IgA antibody is protective in nature, but that is being destroyed by the enzyme produced by the bacteria IgA proteases. Destroy or neutralize secretory antibody IgA, and so allow the bacteria to adhere to the mucous membrane. Otherwise, this IgA antibody does not allow the bacteria to adhere. Lactamases, penicillin destroying enzyme produced by the Staph aureus and Neisseria gonorrhoeae, and these are just destroying the penicillin because of production of penicillinase and sphalosporinase enzyme. Phospholipases cleaves lipids in the membranes, Clostridium perfringens, and gas gangrene. The 10th is antiphagocytic factors. These just avoid phagocytosis by the host macrophages. These are the capsule or the glycocallis secreted by the some bacteria around the cell wall which prevent phagocytosis and opsonization, the process for the killing of the bacteria. Examples are strep pneumonia, Klebsiella pneumonia, Haemophilus influenza, and Neisseria meningitis. Some strains of the same bacteria have no capsule or the glycocallis. Immune system can overcome this deficiency and they are capable of destroying these bacteria. So here one can see the capsule or the glycocalyx. The capsule is this and the glycocalyx is around that. So by these the macrophages and the other neutrophils are not able to engulf it or to catch it and to eat it. And the capsule or glycocalyx is the resistance in eating the bacteria by the macrophages. Here again one can see the capsule that the that the macrophage try to just catch it and phagocytose, but that is being blocked by the presence of this capsule. Bacteria cannot be engulfed, and this is how they can evade the phagocytosis. The antiphagocytic factors continued. M protein of the strep pyogenes and streptococcus are antiphagocytic. It binds fibrinogen from the plasma, which blocks complement binding to the peptidoglycan layer to prevent phagocytosis. So the complement will not be able to act and to phagocytose these bacteria. Protein A, a secreted product of the Staph aureus, binds to Fc portion of the immunoglobulin IgG, so prevent phagocytosis. Normally, bacteria activates classical complement pathway. Phagocytosis occurs after binding of the opsonized bacteria to receptor for the Fc portion of the IgG or C3 regions. Protein A is anti-complementary, so does not allow the complement to work and to cause the phagocytosis. Here one can see the Staphylococcus aureus. It have got the different proteins, protein A and the other proteins, which does not allow the antibodies to engulf the bacteria. So the bacteria has a protein A, which is the hindrance in the engulfing or the phagocytosis of the bacteria, as one can see in Staph aureus. Similarly here, the soluble M protein that do attach to the monocyte and these neutrophils, and just it helps to avoid the phagocytosis, as these proteins are antiphagocytic in nature, and the macrophages and the polymorphs are not able to engulf the bacteria because of the presence of this M protein and protein A and other proteins secreted by the bacteria. Then vaccines in cell wall of the Mycobacterium tuberculosis that resist the digestion by the macrophages. The other mechanism, the rapid turnover of the pili to shed bound antibodies. Microbes move to the tissue through transcytosis. By products of the growth, acid and gas, the bacteria can enter into the tissues. Resistance to the antibiotics, so antibiotics not able to destroy the bacteria. Salmonella typhi and some of the paratype organisms carry a surface antigen, VI antigen, thought to enhance the virulence, and the bacteria are capable of getting enter into the tissues. Here one can see the waxes in the cell wall of the Mycobacterium tuberculosis. Due to that, mycolic acid and all liporabinomanan and the other waxes by which the bacteria are just not able to be engulfed by the macrophages and neutrophils. And here the rapid turnover of the pili just move on and put their tips on the receptors, and so they move to the on the host cell, and ultimately they can get enter into the transcytosis. Is a procedure by which the bacteria can cross the cytoplasm of the host cell, or that can pass from the gaps between the two cells of the host, and this is called transcytosis. This is nanocarrier aided mucosal delivery of the biomolecules. So receptor mediated transcytosis. Transcytosis means crossing through the cytoplasm of the phagosomal escape. This is the 11th parameter. Bacteria are killed by taking into phagosome, then fusion of the phagosome with the lysosome, which bombard, and now lysosome degradative enzymes kill the bacteria. Some bacteria lies out of the phagosome of the host cell and grow in the cytoplasm of the host cell. Examples are the Shigella and the Listeria. These block fusion of the phagosomes with the lysosome, so prevent delivery of the degradative enzymes and can survive in the phagosome. Organism may be resistant to the degradative enzyme of the lysosome, and if fusion with the lysosome occurs, the enzymes cannot destroy the bacteria. So here one can see the natural process, usual process. The bacterium enter through the receptor, and the phagosome just take it inside the bacteria, and the lysosome by its weapon just destroy the bacteria, and these phagolysosomes are formed, and ultimately bacteria out by the exocytosis. This is just because of the fusion of the phagosome with the lysosome. But here one can see the bacteria are not trapped by the phagosome and not attacked by the lysosome, and phagosome fusion occurs and does not trap the bacteria inside as it is here, so the bacteria escape. The 12th is intracellular survival of the bacteria, surviving within the phagocytes and the neutrophils. This allows the bacteria to hide from the antibodies and the host immune response. Mycobacterium in cells is protected from the neutrophils and the macrophages. Chlamydia and the other intracellular bacteria, these are also avoided and protected from the neutrophils and macrophages. Here one can see the bacteria intracellular pathogen, and through the endosome or phagosome get entered into the cytoplasm of the host cell, and here it prevents the fusion with the lysosome, or they just escape in the cytoplasm, or they can survive within the phagolysosome comfortably, and these are not killed by the routine. So this is called intracellular survival of the bacteria. Here is the thought of some artist, and what his thought is that from where the water is coming and then coming down, having the blue color, and ultimately coming out as the green color. That is the thought of an artist. Now, 13th parameter is antigenic variation. Alteration of the surface antigen like protein, LPS, capsules during infection, so there is avoidance of the specific immune response like antibodies which is directed against these antigens of uh, protein, LPS, capsules. Examples are the pathogenic Neisseria brulee, O and H antigen of the Salmonella, O and E antigen of E. coli, O antigen type 1 and type 139 Vibrio cholera, turning pili on and off or switching to the new piles. So this is the process by which one can avoid the phagocytosis. Here one can see the system is on and the antibodies is capable of just engulfing this. But here if the system is off and stop and antibody not effective and target not expressed, antibody is not able to recognize the target and it is there. Here again, if the system is on, antibody less effective and which is very effective and can just get the bacteria and to kill it. But here, if the system is off, the antibody is less effective and the target has decreased expression and antibody cannot get hold of the bacteria to kill it. The 14th parameter is antigenic mimicry. Mimicking means the host molecule. The mimicry means mimicking the host molecule. It is just like example of the caps. If one have used many people wearing the red and green caps, and it has been asked that the green caps should be spared and the red cap people should be killed. So these bacteria, instead of the red cap, they do wear the green caps to avoid the killing by the macrophages. So that is called antigenic mimicry. Pathogen can cover themselves with molecules that resemble normal host or the self molecule, which the body resistance or the body macrophages are not going to kill. Look like the self antigen. Streptococcus pyogenes has a capsule of the hyaluronic acid also made by the host tissue. So the macrophage just think that it is the hyaluronic acid of the normal body tissue. Salicylic acid capsule of the group P meningococcus. The 15th parameter is iron requirement. Bacteria require iron for the metabolism and the growth, and the requirement is 0.4 to 4 micromole per liter. Blood is a rich source of iron, but not free in solution, so not readily available to the bacteria. Iron in blood is bound either to the hemoglobin in the erythrocytes or to the transferrin in the plasma. Some organisms produced iron-binding molecules or the factors which are called siderophores. These compete with the host for the iron bound to the hemoglobin, transferrin and lactoferrin. Binding power of the siderophore for the iron is very high and they can snatch the iron bound to the transferrin and the lactoferrin in the host body. Here one can see the bacterium, and here is the bacterium receptor, and here the bacterium receptor have got the siderophore which is capable of catch the iron. Similarly, in gram-negative bacteria, there are molecules which are enterobactin, salmonellins, sidrocalins, and enterobactin. These molecules of the gram-negative bacteria are capable of getting hold of the iron from the host and then getting transferred through the pores into the bacteria and to maintain the function of the bacteria. Similarly, in case of gram-positive bacteria, Staphylococcus A and Staphylococcus B, they are capable of getting the ferrous iron and transporting inside the Staphylococcus bacteria. Similarly, in the fungal pathogens, the molecules capable of taking the iron inside, that is fusarenine C, and the other molecule ferricrosins, these are capable of taking the iron from the host and getting inside the fungal cell to maintain its function. So the type of the bacterial cytophores are enterocalin, enterobacterin, catechols, phenolates, that is different for the different bacteria produced by the Escherichia and Salmonella species. The Salmonella mutants unable to synthesize enterocolin, which are not able to synthesize enterocolin, they lose their virulence. Injection of the purified enterocalin to these mutants restore their virulence and they become virulent. So, siderophore production by the pathogenic bacteria is an important virulence mechanism. Similarly, iron in milk and other secretions like tears, saliva, bronchial mucus, bile, and gastrointestinal fluid is bound to the lactoferrin. Some bacteria have the receptor for iron-binding proteins like transferrin, like Neisseria species, which can take the transferrin directly from the body. And here is the thought of a photographer, and he have got that photograph at that critical moment when it is just unbelievable to believe that it could be like this. But considering the thought of the photographer, it's really marvelous. Now, 16th parameter is plasmid. It is extrachromosomal DNA segments that carry genes for the antibiotic resistance known as the R factor. Multiple drug resistance plasmids increase severity of the disease by their resistance to the antibiotic therapy. Virulence plasmids are TTSS in Shigella, Yersinia toxin in Salmonella, E. coli, and anthrax. Enterotoxin production by the E. coli and Staph aureus are plasmid-borne virulence factors. Bacteria can regulate gene expression in response to the environmental cues. And here one can see the plasmid. Here is the bacterial DNA. Here is the plasmid, the circular part of the chromosome. And here one can see the donor nucleotide, and that is giving the gene TRA or the resistant genes and the other genes, and that is just going from this plasmid and to this plasmid of the other cells, that is the recipient's nucleotide. So the genes from this plasmid can go and merge into the chromosome or the nucleide of this recipient to become resistant. So that is a kind of resistance transfer to the other bacteria. Now, 17 is bacteriophages. Classical example of phage-directed virulence is seen in diphtheria. All strains of the Clostridium diphtheria produce exotoxin only. But when infected with a bacteriophage called beta-phage, genes for the toxin production is present in the beta or the other tox-positive corynephrage. The elimination of this phage abolishes toxigenicity of this bacillus, so the bacillus becomes toxic only when it is being infected by the bacteriophage to make it virulent. Also seen in botulinum toxins, Shiga-like toxin, linked to the lysis, staphylococcus toxin, TTSS substrate in the salmonella. Here one can see the bacteriophage. These are just infecting the different bacilli, and here one can see these are infecting the Vibrio and the other comma-shaped bacteria, and here the bacteriophage is infecting and transmitting and transferring its genetic material through the tail to the bacterial cell, and bacterial cell becomes more virulent and infective because of having this genetic material transferred through the bacteriophage. The 18th parameter is motility. Many bacterial pathogens are motile: enterics, Campylobacter, Helicobacter, spirochetes. The motility is crucial for the virulence in some cases. Usual organelle of motility is flagellum. Variants are twitching motility, swarming, sliding, swimming, and gliding motility. As you can see, the bundled flagella having rotation, corkscrew like clockwise rotation, then gliding, and then twitching and sliding. And that is all the movement by which the bacteria are able to move from one place to other to cause infection. So now the summary of all this bacterial virulence is serum resistance, as the serum allow the complement to kill the bacteria. LPS, that is also just avoid the phagocytosis of the bacteria because that is antiphagocytic. Capsule also hinders the phagocytosis. Citrophore also taking part like enterocalin and other, then adhescence of the different bacteria by which they are able to attach to the host cell, that is type 1 pili, type 3 pili, KPF fimbriae, and non-fimbrial aggressive adhescence. All these are capable of causing the adhesion. So the other virulence factors are exotoxin, endotoxin, virulence, surface proteins, and pili. Already I have told you, virulent enzymes and flagella and the other parts of the bacteria which are having capability of increasing the bacterial virulence factor. And that is the end of the lecture. I hope and I wish and I request to subscribe my channel and to like and to share it and to also tell to the others if you like. So if you don't like, if you have any question, please feel free to talk to me or give your comments. And thank you very much for this.